Namaskar. Welcome to EPG Pathshala. I am Pooja Kumari from Department of Zoology, University of Delhi. Today in this lecture, we will read about or study about gene knockout and gene knockdown. By the end of this lecture, you will be able to understand about what is gene knockout, what is gene knockdown, what is the difference between traditional and conditional gene knockouts, what are techniques used for the development of gene knockout and gene knockdown model organisms. For example, embryonic stem cells, polymerase chain reactions, RNA interference. Then gene knockout and gene knockdown model organisms, significance of gene knockout and gene knockdown models. At the end of this lecture, we will summarize the particular lecture. So let's introduce the topic. A gene knockout organism is a genetically engineered organism that carries one or more genes in its chromosomes that have been inoperative or non-functional. These organisms are used in studying the function of particular gene by comparing knockout and normal individuals. Gene knockout organisms are very useful tools in reverse genetics, where gene function is discovered after its identification, cloning and sequencing. The cloned genes are knocked out or their expression is altered, and the resulting phenotypes are studied to find out the function of a gene. Knockout means completely blocking the expression of genes mRNA and no production of proteins, whereas knockdown means inhibiting the expression of a gene's mRNA incompletely so that there is a production of some functional proteins. What is conditional knockout? In conditional gene knockout, specific gene function is eliminated in a particular tissue. It differs from traditional gene knockout techniques as it targets specific genes at specific times rather than being a gene non-functional in all the tissues from the beginning of life. It is useful as compared to traditional gene knockout techniques, as in the later, one mutation of a particular gene may lead to the death of an organism as a result of side effects and prevent studies of the particular genes to be done in an adult organism. This technology enables scientists to study the effects of knockout genes at a specific developmental stage and in one specific tissue. This has a further advancement that sometimes effects of a gene in an isolated tissue is difficult to study. So it is required to study a specific gene being inactive in one tissue and remain functional in other one. Now, what is knockout model organism? So first of all, let's talk about gene knockout mice. Gene knockout mice is created by a process in which a normal gene is cloned, which is disrupted by insertion of a marker gene, for example, antibiotic resistant gene, or become non-functional by induction of mutations. This DNA construct is transferred to the embryonic stem cells, where the replacement of normal gene with altered gene takes place. Embryonic stem cells are injected into blastomere embryo, and the mice produced is bred to yield homozygous individuals for the altered gene. In the year 2007, M.R. Capeci and M.J. Evans or Smithens got Nobel Prize in Physiology for Medicine for development of gene targeting technologies essential to the creation of knockout mice serving as animal models for the human diseases. Let's talk about production of gene targeted knockout mice, which involves following steps, like how we can produce these models. First, embryonic stem cells isolation from the inner cell mass of the mouse blastocyst and their culturing. Embryonic stem cells are taken from the inner cell mass of a mouse blastocyst and cultured under the condition on a feeder layer or fibroblast or in the presence of leukemia inhibitory factor, so that stem cells can grow and remain pluripotent, so that they can later, if required, differentiate into different types of cells, example myocardium, myoblast, blood vessels, nerve cells, germ cells, etc. Second one is introduction of a mutant or disrupted genes into the cultured embryonic stem cells and their selection. Various transfection techniques are used for introduction of cloned DNA containing desired gene into embryonic stem cells in culture. The introduced DNA is inserted by recombination into the chromosomal DNA of a small number of embryonic stem cells. The insertion constructs which is introduced into the embryonic stem cells contain 3 genes, the target gene of interest and 2 selection genes, for example NEOR, which confers neomycin resistance, and the thymidine kinase genes from herpes simplex virus, which confers sensitivity to gangliyovirus and cytotoxic nucleotide analog. The construct often is engineered having target gene sequences disrupted by the NEOR gene and the THHSV genes at the end or at the end of the sequence. Usually, most of the DNA constructs will insert at random by non-homologous recombination as compared to the gene-targeted insertion through homologous recombination. If homologous recombination occurs, only the target gene and the NEOR gene will be inserted. In the case of non-homologous recombination, all the 3 genes will be inserted, that is the target gene, NEOR gene and thymidine kinase gene. As a 2-step selection strategy is used to select the embryonic stem cells that have undergone homologous recombination, and as a result target gene is replaced by disrupted gene. First, the embryonic stem cells are treated with neomycin to select only the recombinant embryonic stem cells as they have NEOR gene, and secondly, the recombinant embryonic stem cells are treated with ganciclovir to select homologous embryonic recombinant cells, because these cells do not have thymidine kinase gene, and non-homologous recombinant embryonic stem cells die because of presence of THSKV gene. Homologous, that is the third point, homologous recombinant embryonic stem cells are injected into a recipient mouse blastocyst, and blastocyst is surgically implanted into the pseudo-pregnant mouse. And the last step is mating of chimeric offspring heterozygous for the disrupted genes to produce homozygous knockout mice. The transgenic offsprings developed by this process is chimeric, as it is composed of cells that are derived from the genetically altered embryonic stem cells or from the normal cells of the host blastocyst. If germline cells derived from the recombinant embryonic stem cells, the alteration will be passed on to the next generation. If the recombinant embryonic stem cells are homozygous for a visible phenotypic marker like the black coat color, and these cells are injected into the blastocyst of mice which is homozygous for white coat color, the progeny will be the chimeric and heterozygous for the knockout mutations. These progeny individuals are mated with each other to get the homozygous offspring for the knockout mutation. Now let us talk about gene knockout in yeast. Gene knockout yeast can be developed by using PCR, that is polymerase chain reactions based strategies. On the basis of known sequences of the genome, PCR primers are designed. Artificial linear DNA deletion constructs, that is target vector, is designed and amplified using PCR primers. The DNA deletion constructs, that is target vector, has a sequence of the interested gene which is disrupted by selectable marker. In this example, the selectable marker being used is a DNA fragment containing the CanR, that is kanamycin gene, that confers resistance to the G418. As the coding region of the gene of interest is disrupted by BiCanR gene, this altered gene can no longer code for its protein and becomes non-functional. This linear DNA construct is transformed into the yeast cells, and G418 resistant yeast colonies are selected. This linear piece of DNA is enabled to replicate on its own, so it can be sustained in these cells if it would insert in the yeast chromosomal DNA. The linear DNA constructs can integrate in the yeast chromosome either by homologous or non-homologous recombinations. homologous recombination will result in the replacement of functional copy of this gene of interest in the chromosomal DNA with the non-functional copy of gene on the linear DNA construct. Therefore, homologous recombination event completely inactivates or knock out the chromosomal copy of the functional gene, as the coding region of the gene is replaced by CANR gene. A screening method must be used to differentiate between non-homologous and homologous recombinations. In this case, PCR is used as a molecular screening method. DNA is isolated from transformant yeast cells and PCR is done with the help of specific primers. 4 PCR primers are used, 2 from ends of the gene and other 2 within the gene. If gene is still present, it will produce DNA fragments of predictable sizes. If gene is deleted, no PCR products are going to be observed. Further, to confirm the deletion of gene, PCR primers specific for the CAN-R DNA fragments are used. If there would be deletion of the gene, predictable size of fragments corresponding to the CAN-R DNA fragments will be produced. Yeast is generally haploid, so knockout yeast will not have a second functional copy of the gene, as in the case of diploid organisms. Therefore, the new mutant gene or cell will have a defect in the specific function of the knocked out gene. In knocked out gene is essential for the viability. The new mutant cells will die and no G418 resistant colonies will be recovered. Gene knockout approach is very efficient in yeast, as homologous recombination is quite common in this organism, whereas in other organisms in which homologous recombination is rare, this method would need modifications to get positive results. Let's talk about gene knockout in Mycoplasma genitalium. Mycoplasma genitalium is a bacterium which is known to survive without host and has one of the smallest genome, having about 500 protein coding genes. Transposons are used to make the gene knockout Mycoplasma genitalium. It is known that transposons are capable of moving from one place to the other place of the genome. If they get inserted in a gene, it is going to disrupt its function and gene becomes non-functional. About 2000 transposons insertions were generated, characterized, and their position were assigned to the annotated genome. If transposome inserted into the essential gene, it results in non-viable bacteria. In this way, at least 100 essential genes were identified which are required for the bacterial cells to survive in the laboratory conditions. The goal was to identify minimum number of genes so that an artificial cell can be created. Knockdown model organisms, and what is knockdown? And it can be done by RNA interference. So, RNA interference is a normal cellular process in eukaryotes in which small regulatory RNA molecules silence gene expression in eukaryotes. It is known that RNA typically exists in single-stranded form, but unusual double-stranded RNA in the cells initiate the process of RNA interference. There are different sources of double-stranded RNA molecule. For example, some of the double-stranded RNA molecules are encoded by genes. These genes encode single-stranded RNA molecule, but complementary in different regions of single-stranded RNA molecule result in the formation of hairpin, which is double-stranded. Cellular proteins binds to the double-stranded RNA and cut it into small pieces. Slicer, a protein, binds to the short double-stranded RNA, unwinds one of the 2 RNA strands and discard it. The remaining short single-stranded RNA along with the slicer protein pairs with the complementary single-stranded RNA, and this new cellular RNA is the target of the RNA interference process. Due to pairing, either the translation of the mRNA is repressed or it is cleaved by slicer and pieces are degraded. In the both cases, the mRNA molecule becomes non-functional. There is a protein formation by the mRNA and the gene expression of the gene has been silenced at the translational level. Alternatively, an engineered gene can be used to produce a hairpin RNA transcript with double-stranded portion that can trigger the RNA interference process to silence a specific gene target. RNA interference can be used for silencing the gene expression, and it is very specific because it depends on the complementarity of small regulatory RNAs and target mRNA. RNA interference has been used as a desirable technique to knock down or knock out the expression of genes in various eukaryotes, including Caenorhabditis elegans, Drosophila mouse, and plants. In this technique, the gene is transformed into the organism, which is transcribed into RNA and initiates the process of RNA interference. The gene is designed in such a way that base pairing occurs within the gene itself to form a hairpin structure called a short hairpin RNA. RNA interference is initiated by the double-stranded part of the hairpin structure. RNA interference approach has been used in Caenorhabditis elegans, either knock down or knock out the expression of about 20,000 protein coding genes, and resulting phenotypes were characterized. Now let's talk about knockdown mice. Transgenic mice with reduced expression of target genes has been created. The procedure includes cloning of a target sequence as an inverted repeat, which is separated by short spacer sequences. Transcription of this gene construct results in the formation of RNA transcript, which forms a short 90 to 21 base pair hairpin RNA, small for short hairpin RNA, due to intramolecular complementarity, which results in intramolecular base pairing. Pronuclear microinjection or viral vector like lentiviral vector is used to introduce this gene construct into mouse embryonic stem cells. The produced short RNA is targeted by the animal's dicer and risk proteins into siRNA to reduce the expression of target genes. The same technique has been used to create a variety of knockdown animals, including cows, pigs, goats, frogs, and rats. It has been found that reduced expression of the target gene might impair the animal growth, so expression timing of short RNA would be controlled in the transgenic mice. A blocking sequence is inserted into the DNA construct between the sequence encoding the short RNA and the promoter that controls the expression of hairpin RNA, which results in the conditional knockdown. RNA polymerase termination signal is present in blocking sequence, and it has a marker gene such as GFP, which encodes green fluorescent protein under the control of its own promoter. Two LOXP sites in the same orientation are present on both sides of the blocking sequence, and after induced expression of Cre recombinase blocking, sequence is excised. Expression of the hairpin RNA is restored after removal of blocking sequence, and target gene in the mouse genome is downregulated. Let's talk about the significance of gene knockout and gene knockdown approach. Gene knockout animals, for example, knockout mice, are being used as model systems to study a wide range of processes in mammals, including development, physiology, neurobiology, and immunology. They are used to study numerous human disorders from sickle cell anemia to heart diseases to many different types of cancer. Gene knockout organisms enable to monitor the effects of a particular gene, example obesity, heart diseases, arthritis, Parkinson's diseases, etc. Knockout organisms have been used to characterize genetic regulatory reasons to establish link between mutant phenotypes and particular transcriptional units. This may lead to the discovery of the next generation of therapies for curing numerous diseases based on novel targets from the human genome. So, with the aim of to study human diseases, the National Institute of Health initiated the Knockout Mouse projects in 2006 with the goal of producing knockout mutations in as many mouse genes as possible. This project has been further expanded to the North American Conditional Mouse Mutagenesis project and is working together with the European Conditional Mouse Mutagenesis project to produce at least one knockout mutation of about 20,000 genes in the mouse genome. Using knockout mice, the cell cycle regulating genes and cancer causing genes have been identified. RNA interference has also been used to silence the expression of genes that are aberrantly expressed in cancer cells. RNA interference methods have been used successfully to find out gene functions in various organisms that are usually difficult to manipulate genetically. For example, mosquitoes, trypanosomas and mammalian cells. When these cancer-related genes are silenced, the cancer cells lose their ability to form tumors when introduced into the mice. The nematode Caenorhabditis elegans used as a model organism to understand programmed cell death. With the help of a gene deletion approach, a yeast knockout project is completed in which each gene has or is deleted or become non-functional by replacing with a mutant form. Using this approach, essential genes have been identified as their deletion leads to non-viable cells. Non-essential genes have also been identified in this project as their deletion results in viable cells. This approach also helps in assigning function to a particular gene and therefore is an important tool in reverse genetics. Consequently, genes involved in events of cell cycle, meiosis, DNA replication, transcription, translation to name a few of have been identified. There are certain limitations of the knockout or knockdown methodology. The experimental success is low as many knockout mice die at embryonic stage before they can be used as model organisms for the experimentation process. It is very cost effective and increased cost is required with genetically altered organism care. Ethical issues and objections are associated with the use of embryonic stem cells. Another problem is the deferred function of a gene in embryo and developing embryo. Knockout or a knockdown have a specific gene which may be having different observable results in model organisms and in humans. For example, mutation in p53 genes associated with the cancer and tumors. Let's summarize this topic. A gene knockout organism is a genetically engineered organism that carries one or more non-functional genes in its chromosome. In this organism, there is no production of proteins encoded by the particular gene. On the other hand, in a gene knockdown organism, there is reduced expression of the gene and it is not completely non-functional. Various molecular techniques have been used including embryonic stem cell techniques, PCR and RNA interference to create knockout or knockdown model organisms. For example, gene knockout mice, gene knockout yeast, gene knockout Mycoplasma genitalium, gene knockdown Caenorhabditis elegans, Drosophila to name a few. Gene knockout and gene knockdown model organisms are important to understand the function of a particular gene in reverse genetics to study numerous human disorders. This may be helpful in development of therapies for curing various diseases based on gene analysis of human genome. Thank you.